Hello, everybody. It's a great pleasure to be here, and I really appreciate the invitation. Sorry I can't be with you in person today, but I hope I can convey some interesting story on some of our research on metabolic disease, metabolism, aging, and cancer. So as we just look at the nature of what the KAIX program is here, I think that this is a very important area to be exploring and to develop. The theme of this presentation is deconstructing risk factors for colon cancer and inflammatory bowel disease, IBD. The subtitle here is Unlocking a New Therapeutic Approach. That is, these are very difficult diseases to treat. Colon cancer is one of the most challenging diseases to treat, and so coming up with new therapeutic goals is very important in this increasingly challenging disease. So if we go to the next slide, when we talk about risk, why is cancer risk and IBD risk? What is it, and why is it important? Just to go back, because our approach to trying to understand disease mechanisms and therapeutics is to focus on the nuclear receptor superfamily. This is kind of shown here as a schematic in terms of the use of the Rosetta Stone, which was the basalt tablet that was the tablet that translated Greek and Egyptian hieroglyphics and Demontic language into this one entity. And so the nature of using the Rosetta Stone here is that the communication between different parts of the body and physiology are kind of embedded in the way in which the stone is crossed here. So in the upper part, we are looking at the receptors such as TR or GR or RAR and thyroid hormone, glucocorticoid receptors. These are the nuclear receptors up here. These are the DNA binding sites for these particular receptors. So when you translate what the receptor sees now to something that goes down to the transcriptional template, and then ultimately is regulated down here by the hormones, such as glucocorticoids or thyroid hormone or estrogens or androgens. And the Rosetta Stone is what brings these all together. And the way that we can more effectively visualize this is looking at the superfamily. It began in 1985 with the first isolation of the glucocorticoid receptor, which provided a molecular template, which is shown on the bottom as a series of domains, and all nuclear receptors basically have this exact same domain structure. And at the time, since we were isolating a steroid receptor, we thought that we would be getting a small family of steroid receptors, ultimately 5 or 6 of steroid receptors. But in the end, what we thought we were going to get, there were 42 additional nuclear receptors that were unknown, and therefore 42 novel physiologic pathways that were not known. And that's what became the superfamily. And I'm not going to go through too much detail on this, but thyroid hormone, vitamin D, retinoic acid, glucocorticoids, these are all very potent regulators of metabolism and development. Glucocorticoids is the fight or flight response. It puts a lot of glucose in your blood very instantly. It suppresses inflammation, suppresses pain, so you can fight and not feel pain. Has profound effects in thyroid hormone. It's about thermogenesis and therefore also about energy control. And many of the receptors are about metabolism. I'm going to talk about specifically One is called FXR, which is the bile acid receptor, and bile acids are basically a form of steroid that is produced in the liver, and it's collected in the gallbladder, and I'll show you a little bit about its properties in a moment. But now we have a superfamily here, and it's up to us to both deconstruct it and then to use these discoveries for new types of therapies. So this is deconstructing colon cancer risk, revealing FXR as a therapeutic target, and that's really the principal goal of this talk. And why am I speaking of cancer risk in general, and why is this important? Let me briefly say that the discovery of the bile acids initially goes back to Adolf Windaus, and he won a Nobel Prize for his discovery of bile acids and their similarity structurally and chemically to vitamin D. So this was a complete surprise. Vitamin D is completely unexpected to be related to a sterol. And this opened up a whole new field because there are many types of bile acids in our bodies, at least 30 to 35 different species of bile acids. And what we learned, and that was the concept for decades and decades, and that bile acids were surfactants that allowed lipid to be absorbed into the body. And while that is correct, what wasn't appreciated is that bile acids are actually hormones. And that's what our discovery of the first bile acid receptor, FXR, in 1995 changed the field of bile acid physiology into one that is not just about transporting nutrients around, but rather is operating through a nuclear hormone receptor. That means changing genetic patterns throughout the body. Very different way to think about how this system works, and open up an entirely new approach to thinking about bile acids, what they do, and how they might be used to study disease mechanisms and therapy. So getting back to risk, there are a lot of factors that are linked to cancer risk, and you'll probably know many of these. For me, the most dominant ones are sedentary behavior, that is not using your muscles and other parts of your body to burn energy. So sedentary behavior and high-fat diets are the most common drivers of disease mechanisms, including colon cancer and IBD. The consequence of high-fat diets and sedentary behavior is links to the obesity epidemic. The obesity epidemic has been ongoing for the last 33 decades, at least, and it's also moving to a younger and younger group. And so not only is the epidemic going, but it's actually growing because of its increasing number of people that are becoming overweight or obese. High-fat diets lead to intestinal inflammation. This is a risk factor for cancer. Family history. If someone in your family has had a disease, your risk immediately goes up because it may have a genetic underpinning in your lineage. And then age. Age is a risk factor. The longer you're on the planet, the more chance you have of getting cancer or inflammatory bowel disease or a number of other different types of diseases. And what does disease risk look like? Well, one simple way is a geopolitical view of a risk. And in the U.S., we can see that Colorado is the lightest state in terms of the weight of the average weight of people there versus Mississippi, which is the heaviest state. And the heaviest state also has, as the fattest state, also has the highest obesity population as well. And the same map looking at colon cancer leads to the same two states being Colorado the lowest risk if you live there, and Mississippi the highest. So there is a geopolitical aspect to cancer as well. So we think of risk not just being just the high-fat diet, but where you live can be a risk. And so understanding this, I think, is very important because the word risk does not express a mechanism. And if you don't have a mechanism, it's very hard to come up with a therapy. So in context of colon cancer, there's something that's called a Vogelgram that comes from Bert Vogelstein, and he proposed this simple scheme here where you have a normal epithelium in the gut, and with a mutation, what's called the APC min gene, anomalous polyposis coli gene, that you can get something called an early adenoma. You get proliferation. So this gene is linked to approximately 75% to 80% of colon cancer patients have a mutation in this specific gene. And it leads to proliferation in the crypt of the colon, but it's not cancer. If you have a KRAS mutation, in addition, then you get what's called late adenoma. So there's a progression here, but it's still not cancer. If you get a P53 mutation, then you get cancer. And this is the carcinoma that's shown here with P53. And then because the proliferation rate is up higher, you now have a bigger chance for getting metastasis. And these are additional mutations that occur over this time span. And time span can be sometimes 20 to 30 to 40 years. People don't know what they have for quite some time in this disease mechanism. And so it's been challenging both to diagnose early on, and no matter what, it's challenging to treat. And what is happening in this lineage is a little bit perplexing. It's simplified in the diagram as 1 mutation, 2 mutations, 3, 4, going in that order. When we began this project, I thought that was the way it works. Turns out that's not true. This is a very simplified diagram and never really had been re-examined since the original article back in 2003. And I don't really agree with this particular diagram anymore. And so there's something that I call Pandora's box. This is the thing that holds in, the Pandora's box holds in various types of maladies and problems, and then when you open it up, they come out. And so what is actually inside the disease mechanism? So just to illustrate this in a simple way, if we look at these cogs that are interacting with each other, there's a hereditary cog, and that's if you have something like Lynch syndrome, something in your family, your risk is higher, and that begins to put pressure on growth. There's an environmental component, which, like the Western diet, the high-fat diet, that's also a key component here. And then the key thing for cancer is increased replication, and so somehow the risk with the familial link and the environmental link have to put pressure on replication to increase. And that is quite a bit of a challenge. And so, but all these components are interacting with each other, and this is just a way to break down the nature of the system. And what are the links between H, E, and R? Hereditary, environment, and replication. And that's Pandora's box. We don't really know. And I want to use this as a transition, talk a little bit about the core theme here, the bile acid receptor FXR, which I mentioned earlier. And what you can see in this diagram here is that bile acids, there's over 30 of these species. They're synthesized in the liver, then delivered to the gallbladder, and when you eat a meal, they're released into the intestine. The nature of the bile acid receptor, the organs that are shown here, kidney, the adrenal gland at the top of the kidney, the liver, gallbladder, the intestine itself, the pancreas, these are all expressing very high levels of the bile acid receptor, FXR. So these are FXR target tissues. What does that mean? This is just a schematic of the FXR receptor interacting with a ligand. This is a classic bile acid that would bind to FXR, chenodeoxycholic acid, and then act now, as we believe, as a steroidal-like hormone, activating genetic programs in the intestine, liver, and other organs. We can then begin to explore this in some specific ways. What I want to specifically point out here is my lab created a form of a ligand for FXR that's a synthetic compound. This is a chemical that we made in the laboratory. It binds selectively to FXR, and in particular, this compound only acts in the colon. It does not get into the body, does not get into the bloodstream. And because of that, we're going to look at what is the impact of activating intestinal FXR with this drug called fexaramine, FXR amine. That was the title. So we call it FEX. And we'll kind of use this to explore a little bit about the nature of the signaling pathways. It's called the farnesoid X receptor, and that's because farnesyl is one of the direct synthetic precursors to cholesterol. This is in the cholesterol synthetic pathway from mevalonate going forward. And it's also very, very similar to juvenile hormone, the molting hormone in flies. And I should mention that the FXR receptor in humans is very closely related to the ecdysone receptor in insects. And juvenile hormone in insects, it controls molting. And so a lot of plants make juvenile hormone, and they do that to prevent insects from eating them. That juvenile hormone in the fly is acting on the fly equivalent of FXR, which is the ecdysone receptor. And FXR and ecdysone bind to the same exact sequences as well in very similar ways. So the point of this slide is to show you the complexity of the generation of bile acids, as is indicated here, both in what's called the neutral pathway and the acidic pathway. And this represents hundreds of millions of years of evolution to create this complicated biosynthetic theme. All these are steroidal-like hormones. So there's a lot of importance if they're going to have this kind of a pathway here. And we're very interested in the pathway, number one. Number two, what was not appreciated is that the synthetic rate at which you can process and produce bile acids has certain boundaries, and it can only produce so much in a particular unit of time. But this is the nature of the complexity that you have to deal about when you're talking about bile acids in general. So here are the questions we want to ask. For bile acids, they're linked to inflammation and colon cancer and IBD, as you'll see. What are the unsolved questions? So how does FXR impact intestinal homeostasis? This is key. How do bile acids modulate intestinal physiology? Pretty straightforward, but not actually when we get into it. How does high-fat diet promote intestinal inflammation? How does high-fat diet accelerate cancer progression? And is FXR a therapeutic modulator or a therapeutic target in colon cancer and IBD? And so, and we're going to keep looking at it in the form of hereditary, environmental, and replication components. Now, first surprise we got that disrupts the Vogelgram is the nature of the that cascade that begins with APC min mutation. Now, we have mice in this study that have an APC min mutation or not. This is a normal mouse, a wild-type mouse on chow diet, and you can see that the size of the colonic epithelia cells here in the crypt is very long. If you put these animals on a high-fat diet, this is a different angle, but this is the arrow now that you get. And you have a much shortened epithelial cells here. Just the diet is having a structural change, most likely due to inflammation, that is really shortening the crypt. And this is what happens pathologically, and you can see this visually in these slides. And the question is, do these visual changes have some kind of impact on intestinal function? And so diet's changing structure. What is it doing about function? And that's the next question that we want to address. And here we are using APC min mice, either on a normal diet or on a high-fat diet, as is shown here. And you can see on a normal diet, you have the adenoma, which is expected. But unexpectedly, this is the one that's not cancer. But in this case, you're going from adenoma, which is that first step that we saw with just the APC min mutation, to adenocarcinoma. So you have jumped over the RAS, MYC, P53, the whole, that slow progression of a change. We have leaped, and those changes occur over 20 or 30 years, we have leaped into immediately using diet, which jumps over all that and creates adenocarcinoma. And so this is unexpected. It's the first time that this has really been seen. And now you begin to understand the link between diet and cancer risk that we didn't fully appreciate. And you can actually visualize it here. So when you try to consider what this questions that this raises is, how does disruption of the villus, the crypt villus as we see here, how does that actually affect gut physiology and intestinal function? And if we just look a little bit about what is happening in this process, it's really surprising. And if you look in wild-type mice and look at the serum bile acid level, all the bile acids you make, 95% get resorbed, and they're in your body going everywhere, to the spleen, to the heart, to the brain, the muscle. They're just being distributed everywhere like any other steroid hormone. So that's your bile acid pool on a normal diet. On a high-fat diet, you more than double the pool. Now, in physiologic terms, doubling a physiologic factor is rather dramatic. Think of it as doubling your salary, or doubling your weight, or doubling what your bank account is. Oops. Interestingly, in the APC min mouse, and this is a haploid. APC min mutations are haploid from the mother, and what is happening here is in the normal diet, just the mutation is driving up the bile acid pool. The bile acid pool generation in the liver, due to this change in APC min, is so APC min as a proto-oncogene is driving an action in the liver, specifically focusing on bile acid generation. That was the next surprise. If you put it on a high-fat diet, you get this, gargantuan, tremendous increase in the bile acid serum pool. So this now leads me, and I was very, taken by these results, to begin to think of why is the bile acid pool so dynamic and what is happening here. This is the beginning of trying to deconstruct what the diet is doing besides changing the pool. And one measure to look at it is looking at something called leaky gut, leaky gut is about intestinal permeability. If you have a leaky gut, typically linked to inflation, inflammation, inflammation that bacteria and other components and fragments of bacteria can get into the bloodstream because it's leaky typically when there's inflamed intestine. So on a normal diet, you have very tight, no leakage, basically. This is looking at a fluorescent dextran getting into the blood, very easy to detect and quantify. On a high-fat diet, you can see that you start getting a leaky gut even in a normal mouse, putting it on the high-fat diet. So again, you can see the pathophysiology here. If you look again at the APC min, just the APC min is creating a very leaky gut. This is intestinal permeability that is really quite dramatic. And you put it on a high-fat diet, and that almost doubles. And so there's a process that is going on here that one has to think about in a particular way. For this specific reason, I named this process physiologic convergence. This is a term that basically I made up to show parameters such as diet and genetics as interacting with each other in some convergent way, typically. And so the synergy that you get between these is very important. And the inverse of that is pathologic convergence. So if you have a high-fat diet and a mutation, you then get the mutation and the high-fat linking up, each of them independently on their own able to drive up, for example, bile acid production and leaky gut. But when you put them together, you get this synergy. And so that's pathologic convergence. That is now driving these events. And so getting back to risk again, we're beginning to see what the mutation is causing in a risk and what the diet is causing in a risk. And remarkably, much of it is the same. So that's an interesting thing to puzzle through. And so now, I mentioned bile acids. There are at least 30 plus species of bile acids. What I just want to say here is not all bile acids are equivalent. You saw that big panel of components. And so in particular, taurbeta-muricolic acid, so this is a rodent specific bile acid, but it's known by many labs to be a relatively inflammatory bile acid because it causes inflammation. Now again, because you're an inflammatory bile acid, it's not telling you how it's causing that inflammation, but this is an inflammatory bile acid, and in particular, it's increased, as you can see here, on a high-fat diet more than double. Now the scales are going to change a little bit here. That's why I get this. This is what you just saw, but with the APC min mutation, and these scales are now the same, this is what you have on a normal diet, and this is what you get on a high-fat diet in terms of taurbeta-muricolic acid change. This is a 50-fold change in this one species of bile acids. So the point being is that when you're making all these different bile acids, they're not being made in equal exact amounts. They're being made in different ways, and we know very little about the differential aspects of this synthetic pathway that would drive, for example, the creation of taurbeta-muricolic acid relative to chenodeoxycholic acid or deoxycholic acid. And this now also changes our view of not just the risk factor, but the underlying mechanisms that are drivers for this process. Is taurbeta-muricolic acid important? I'll show you in a minute that it is, but before we get to that, I wanted to make a slight transition because I've talked about intestinal inflammation driven by the high-fat diet, and I wanted to link to another type of intestinal inflammation that has in itself a number of different aspects in its evolution, and this is inflammatory bowel disease. And inflammatory bowel disease is based on a series of cytokines that are produced by innate lymphoid cells that are called ILCs, and in this context, just a little bit of note in terms of one of the major observations that we've made is IL-17, which is a known potent inflammatory cytokine in the gut. In no matter what situation, if we treat the animal in either yellow or green with the synthetic ligand that we have, yellow brings down the inflammation, green brings it down in these different contexts of normal diet, high-fat diet, or chemically induced diet. And so you want to be in this green column if you can. And so what we're saying is giving back and activating the FXR receptor is actually potentially a way to think about in colitis and colitis-induced cancer models this a potential therapy. So just as a summary, if we have HE and R, we get to a process that, and we can categorize these different components. We have genetics, like the APC man. We have the diet, like high-fat diet, that is sort of converging on changing to our beta-uracolic acid, the deoxycholic acid, and other bile acids. We've profiled them all. Acting on FXR, and these altered bile acids almost universally are antagonistic to FXR activation, and that reduces FXR. Knockout of FXR is also very inflammatory, promotes leaky gut as well. And so there's a lot of different things in terms of the driver mutation and the diet, and the bile acid pool composition now and function that are coming into play as we use FXR as our guide to sort of unlock the pathway. And at the same time, I think that this opens a door to think about how we might be able to intervene by reactivating FXR activity. So I mentioned a little bit about some of the actions that are being produced in these various different states. This is one that I'll just briefly mention, looking at the production of FGF15 in the intestine. This is one of the important regulatory factors. It's FGF19 in humans that is very highly controlled by the bile acid receptor FXR, and when it's activated, typically during a meal with normal bile acids, then FGF15 is produced and travels from the gut to the liver, and it controls bile acid synthetic processes. So this is a major regulator of bile acids, and what you can see here is in this that on a normal diet and on a high-fat diet, this critical regulator of intestinal function and the response in the liver is very dramatic. And just to point out, I'll just point out here, for example, FGF15 in red, which is the APC main component. That means it's you can't produce FGF15 basically. If you give the ligand, you restore, as is indicated here. And the same thing is true. Basically, where you have red, it's being suppressed. Where you have green or yellow, you are seeing induction. So this is really important. And again, this is rescuing in some very, very wonderful ways, depending on if we're using the adenoma or the adenocarcinoma diet. So the adenoma is the high-fat diet alone. This is with the mutation. And the effects are really quite dramatic. So what about getting back to aspects of cancer? And importantly, FXR, and looking at one of the key features here, which is leaky gut. And if we look at, in the adenoma case, that is not on high-fat diet, so what we see is an APC, basically at no leaky gut with APC min. You have leaky gut, you can see the amount. With FXR activation, you really bring this down. If you look at intestinal permeability, an adenocarcinoma, same thing, just the numbers are larger. You have dramatic intestinal permeability. You're bringing it down with the drug to about 100. This gets you to 75 or so. So this is actually quite dramatic. And in general, this approach says that it might be possible to restore gut integrity in both the adenoma and the carcinoma model. So this begins to let us think about, might this really be a therapeutic? And so we explore this a little more in terms of how FXR impacts both the bile acid pool and function. And this is one of these just very dramatic results. I'll go over here in the adenoma case, which is wild-type mice. There's very little bile acid in terms of what's in your bile acid pool composition, plus or minus the APC min mouse in diet. So there's not much to regulate. If you see what's happening in terms of in the adenoma component with APC, APC is already driving up the BA pool. FEX brings it down significantly, at least by about 40%, not fully. But more surprisingly is in the adenocarcinoma, where we are putting the APC min mutation on high-fat diet, you're getting up to just a gigantic serum pool size. And in that context, you now see FEX-D bringing things down. Keep in mind that people who are obese and have an APC min mutation are going to be likely in a very dangerous state. This suggests that we could lower risk and also by giving us a mechanism, potentially a way of controlling the process. So FEX-D decreases serum bile acids in both states, but look at that. It's just totally crazy in a way. So how does it shift bile acid composition? I'm not going to really go into that in a lot of detail because I can't really tell you. It is one of these challenges now that we have to look at. I suspect that the pressure on the enzymatic pathways cannot be sustained at the level at which the drive is to take in all of this lipid. And so I think the system somewhat crashes and you probably get incomplete products in this process. And so we're very interested in digging into this in much more detail. But just to summarize, you've seen this chart before. And dietary risk now triggers this cascade of events that increases specific bile acid species. This led us to consider whether we could actually use fexaramine, intestinal-specific ligand, to treat these animals in both the APC-min on normal diet and the APC-min on high-fat diet. So these are all going to be APC-min. And I should say that it's not an ideal model because in humans it's colon cancer. In rodents, it's upper GI cancer, and they typically die from blockage. What I didn't mention is the APC-min mice on high-fat diet do get colon cancer, but blockage is a challenge in this system. But either way, if we give FEX-D, we get an extension here from 30 to 50 weeks, and that shows conceptually that a gut-restricted activation of FXR is able to change a gene network. And we can measure that gene network, and we have. We've done the same in these two contexts. We've also worked with organoids, human organoids as well, to look at changes that we can establish gene expression changes by delivering the ligand or not. And so this then led us to, because we have the gene changes, we can take those genes and go into the database for humans with colon cancer and ask if you have the FXR gene expression that we have seen in human organoids or the APC-min mouse, so human and mouse. If you have this gene set, you are likely to have a increased survival probability. And so that now is somewhat translating the concept of pathologic and physiologic convergence and taking away the pathologic state at least far enough to have an impact potentially on survival. And so this obviously opens up a way to begin to think about future studies and what we might be able to do. And to just quickly summarize, I'm calling it bile acids back to the future. It was one of the great stories 100 years ago. And now, flip ahead 100 years, it's entirely changed. The narrative that it was in that era was all about surfactants and other properties directly in a conceptual way that could be used. And people did use bile acids in therapeutic contexts, like ursodeoxycholic acid, for example, but not in any way thinking it's acting like a potent steroid hormone. And so Back to the Future just means that we have to look back to map the way forward. And by the discovery in 1995 of the bile acid receptor FXR, which I should say was also worked on by David Mangelsdorf and Steve Cleaver, two former postdocs in my lab, who went on, in addition to looking at the activation by farnesols, identified that bile acids were also potent activators of FXR. And so I like the idea of these independent scientists, but in the family, really cracking open some important aspects of this work. So looking backward and forward, the goal is to create a comprehensive molecular characterization of the mammalian biome, another term that I'm making. And the initiative that I'm thinking of conceptually is what I call biocarta, building the biome. And the idea here is that bile acids are dynamic. Their chemical composition and functional properties vary across time, diet and genetics and disease. So they're very dynamic. The BA pools respond to external stimuli, food and the microbiome, bile acid species, number of nuclear receptors, not just FXR, but these other nuclear receptors are all good bile acid receptors. This one is an exception. This is a G protein coupled receptor, TGR5, also a good bile acid receptor. So we're learning so much more about bile acids than we ever knew, and that opens the door for this being a major new approach in physiology. And I call this a liquid organ. Because these are hormonal, having all the properties of a regulatory event, but the way in which they are passed from the liver, packaged in the gallbladder, and delivered collectively. I'm calling it a liquid organ as a way, again, to conceptualize the thinking about how this washes through our system and impacts on our body physiology. And so one way to begin to think about classifying these, this is very simple, is taking the known bile acids, let's assume here on the left, and simply stacking up to, are they activators of FXR? Are they repressors of FXR? Do they have or no have any activity at all? Do they impact the microbiome? Are they changing lipid production? Do they interact with vitamin D receptor, TGR5, any of these other receptors that I've already mentioned? How do they impact the APC-Min pathway? And you can just increase the depth of this and begin to deconstruct something that has been around for 100 years, but now in an entirely new way. And keep in mind the networks that are controlled transcriptionally in target cells throughout the body are enormous. And that means that there's in the way that you achieve this benefit is because of FXR and its distribution throughout the body. So deconstructing risk reveals an unrecognized mechanism of convergent pathways that deplete FXR signaling. This suggests that FXR activation by virtue of synthetic ligands, in our case just an intestinal ligand, could dually block the oncogenic and high-fat diet cascade to restore intestinal homeostasis. The current interest in my lab is homeostasis, and enable a new approach to cancer therapy. The goal of the work here is really trying to deconstruct, that is, asking the right questions. And for the postdocs that are here in the audience and the students, asking the right questions is very important in designing and thinking about the goals that you want in your research. In our case, investigating and better defining risk. That is something been around for decades and decades, but it's never really been defined. And this is what it reveals and also what it covers up. Just saying there's a risk factor, you think you know something about it, but it's really covering up a lot of things. So perhaps the oncogene, it wants to be controlled. It looks like it can be controlled. And so it's not necessarily a villain. It's just waiting for something to come along, in this case FXR, with a ligand, to turn it back on, and the oncogene can go back into its place like it had been for decades earlier. And with that, I'll thank all the people that did the work and happy to take questions if I can. All right, thank you.